Hello, welcome to Biology, the scientific study of life. Natural selection, genetic drift, and gene follow can alter a population's genetic composition. Note again the five conditions required for a population to be in Hardy-Weinberg equilibrium. These are five conditions. A deviation from any of these conditions is a potential cause of evolution. However, although new mutations can modify allele frequencies, the change from one generation to the next is likely to be small. Recombination reshuffles alleles but does not change their frequencies. Non-random mating can affect relative frequencies of homozygous and heterozygous genotypes, but by itself usually has no effect on allele frequencies. The three major factors that alter allele frequencies and cause mass evolutionary change are natural selection, genetic drift, and gene follow. Natural selection, as we read, Darwin's concept of natural selection is based on differential success in reproduction. Individuals in a population exhibit variations in their heritable traits, and those with variations that are better suited to their environment tend to produce more offspring than those with variations that are less well suited. We now know that selection results in alleles being passed to the next generation in proportions different from their relative frequencies in the present generation. For example, in our imaginary wildflower population, white flowers C to the power W, C to the power W, might be more visible to plant-eating insects, resulting in more white flowers being eaten, and perhaps red flowers C to the power R are more attractive to pollinators, increasing the opportunity for red flowers to produce offspring. These differences in survival and reproductive success would disrupt Hardy-Weinberg equilibrium. The frequency of the C to the power W allele would decline in the gene pool, and the frequency of the C to the power R would increase. Okay, we will examine the process of natural selection more closely. Genetic drift: flip a coin 1,000 times and a result of 7 heads and 300 tails would make you suspicious about that coin, but flip a coin 10 times and an outcome of 7 heads and 3 tails would not be surprising. The smaller the sample, the greater the chance of deviation from the predicted result. In this case, an equal number of heads and tails. Similar deviations from the expected result, which occur because real populations are finite in size rather than infinite, explain how allele frequencies can fluctuate unpredictably from one generation to the next. Such fluctuations are called genetic drift. Consider the example, this one. Note that in this example, one of the alleles has been lost by an indiscriminate event. That is, it was a matter of chance that the C to the power R, notice that, C allele rather than the C to the power R, notice that, allele was lost over time. Drift tends to reduce genetic variation through such losses of alleles from the gene pool. Two situations that can increase the likelihood that genetic drift will have a large impact on a population are referred to as the bottleneck effect and the founder effect. The bottleneck effect, notice that, a sudden change in the environment, such as a fire or flood, may drastically reduce the size of population. In effect, the survivors have passed through a restrictive bottleneck, and their gene pool may no longer be reflective in the original population's gene pool. This is called the bottleneck effect. By chance, certain alleles may be overrepresented among the survivors, or may be underrepresented, and some may be eliminated altogether. Genetic drift may continue to have substantial effects on the gene pool for many generations until the population is large enough that chance fluctuations have less effect. One reason it is important to understand the bottleneck effect is that human actions can sometimes create severe bottlenecks for the other species. For example, in the 1890, hunters reduced the population of northern elephant seals in California. Notice that to about 20 individuals. Since then, this mammals has been protected, and the population has rebounded to over 30,000. However, in examining 24 gene loci in a representative sample of the cells, researchers found no variation. For each of the 25 genes, there was only one allele. In contrast, populations of the southern elephant seal, a closely related species that was not so severely reduced, show considerable genetic variation at this loci. The founder effect. When a few individuals become isolated from a larger population, this smaller group may establish a new population whose gene pool is not reflective of the source population. This is called the founder effect. For example, the founder effect can occur when a few members of a population colonize the new location. These founders pass through an isolation bottleneck and represent a distinct gene pool with different allele frequencies from those of the parent population. The founder effect probably accounts for the relative high frequency of certain heart disorders among isolated human populations. For example, in 1814, 15 colonists founded in a British settlements on Tristan da Cunha, a group of small islands in the Atlantic Ocean midway between Africa and South America. Apparently, one of the colonists carried a recessive allele for retinitis pigmentosa, a progressive from blindness that afflicts homozygous individuals of the 240 descendants living on the island in the late 1960s had the disease. The frequency of this allele remains 10 times higher on Tristan da Cunha than in the populations from which the founders came. Gene follow. A population may gain or lose alleles by gene follow, genetic additions to and/or subtraction from a population resulting from the movement of fertile individuals or gametes. Suppose, for example, that near our original hypothetical wildflower population, there is a newly established wildflower population consisting primarily a white-flowered individual, C to the power W, C to the power W. Insects carrying pollen from these plants may fly to and pollinate plants in our original population. The introduced C to the power W alleles will modify our original population's allele frequencies in the next generation. Gene follow tends to reduce differences between populations if it is extensive enough. Gene follow can amalgamate neighboring populations into a single population with a common gene pool. For example, humans today move much more freely about the world than in the past, and gene follows has become an important agent of evolutionary change in human populations that were previously quite isolated.